Welcome everybody. I'm Professor Steve Forsyth, and today I want to do a presentation on the diversity of the predicted antibiotic resistance profiles in the bacterium Cronobacter. For those who don't know me, I'm the curator of the Cronobacter Pub MSD database. That's the URL where you can access it, it's open access, and that's my email address if you want to contact me. So today's 19th November 2024, and this is a snapshot of what I've been able to find out based upon whole genome sequences. As I say, I'm the curator for the Pub MSD database. For those who don't know it, it's an open resource. You have typing, you have isolate collection, you have a genome collection, and you can do blast searching, etc., etc., etc. here. If you want to look at video guides, they're listed here as well. And that's the link which you can click on. In the past, I've done in total 7 YouTube presentations. One of the last ones I think was this one where I was analysing 1,000 Cronobacter genomes and I was reporting what I was finding in that diversity. One which I think is very important for people to look at is the issue over identification and so often the misidentification of Cronobacter that's occurred. And I think that's very important for people who, especially who are new to the topic, relatively new, to just pause, watch that video so you understand which papers maybe in the past may not be as reliable as they may first seem. Also to be aware that there are different pathivars and the clonal complex 4 is the one which can be associated very much with infant or neonatal meningitis. These are 2 videos which are linked together because the first one is an American situation 2021-22, and then I did an update video on that. And then finally 2 videos here where I looked at one outbreak over a 10-year period, i.e. 10 years with regard to the techniques, where we had started in the early days with pulse field gel electrophoresis, and then looking at the same strains all the way up through 16S, multi-locus sequence typing, and even looking at CRISPR-Cas arrays. So a wide variety of techniques there, and all these titles should have active links for you to try out. So why am I doing this? Well, occasionally it comes up with what's the antibiotic resistance in Cronobacter, and I decided that I would do a trawl for myself to see what I could find out with all the information I could gather quite easily. And I want to emphasize that I'm just looking at the Cronobacter genus. I have not specifying Cronobacter sakazaki. Cronobacter sakazaki is not the only pathogenic Cronobacter species. So because of that, I would emphasize it. So I am doing the genus, not just the species sakazaki. And in order to do this, I thought it best to look at a very large number of whole genome sequence strains, and I wanted them to be geographically, temporally, location and source diverse. And it's to review the predicted antibiotic resistance diversity and determine their frequency, and hopefully this may help to motivate further investigations into the actual mechanisms of antibiotic resistance in the genus. What I'm not covering is the location of those genes. I'm not differentiating whether those genes are on the chromosome or plasmid-borne or both, etc. And I am not referring to any treatment protocols. This is purely an initial academic exercise. That's it. Is it a limited study? Yes, it's a limited study. I mean, what's representative? I am only looking at what's predicted. I'm not looking at what's been laboratory confirmed. Some countries report sequences more than others, and some sources are sequenced more than other sources. So it is not representative. It is an overview of what is out there looking at whole genome sequences. The number of genomes I looked at were 2,662. And the source was the dates that I took. All that information was the 16th November 2024. The number of individual gene predictions was over 3,000. The number of AMR profiles was 58. I'm going to be presenting those in about 3 tables. And it covered 35 countries in total. Here's the 35 countries, but not all countries sequence and report to the same extent. The top 5 were America with 1,570. China was about a fifth of that. Ireland was half of that again. Germany was less than Ireland, and then half of Germany numbers were Switzerland. So those are your top 5. So it is skewed. I tried worldwide the best I can, but it is a skewed sample. When we purely look at the resistant genes, the genes which occurred most frequently were the BLA beta-lactamase genes. 2,363, and there's 1, 2, 3, 4, 5, 6, 7, 8, 9, at least 9 varieties or variants of the BLA genes which were there. There was also AMP-C, 314. So your beta-lactamases were the most numerous antibiotic resistance genes found by far. Far less than that were colistin linked resistance genes, 155. 3 varieties of MCR. The most numerous was the variant 9.1. There was the 1, 10.1, and I think there's 2, maybe 2 or 3, 11.1 variants. We do have some trimethoprim, and less than that we have various aminoglycosides. Then we have sulfonamide, tetracycline, chloramphenicol, and then quinoline, followed by lesser rifamycin, erythromycin, and macrolides. And then separate, we have the issue about multi-drug efflux pump, which is an issue I need to bring out very early on in this presentation, because what I found is that for some whole genome sequence strains, although the antibiotic resistances had been analysed and reported, the whole assemblies were not available. Most of these were from 2014, and that covered the multi-drug efflux pumps, unfortunately. I said, well, most from 2014, there were a couple from 2019, but any entry referring to the multi-drug efflux pump were from strains for which there are no assemblies available to the best of my knowledge. So with that caveat, we'll move on. When we look at the BLA genes, the most numerous BLA variety was the CSA. Most often the variant wasn't designated, so we have 1,308 not designated. We have 100 designated to variety 1, and just over 540 designated to variant 2. Then we have lesser ones with, we have multiple BLA varieties together, and we have the varieties including trimethoprim, and we have our colistin genes coming in here as well. So a number of strains did have more than one type of BLA gene in it, and the colistin variety 10.1, I think this was the only occurrence. I looked up that individual strain. It's the clinical one, 2019 China, and it was Cronobacter sakazaki. I was also interested in this one in particular, just highlighting it just out of curiosity, because fluor is meant to be resistant to an antibiotic which is used in animals, and that was again a sakazaki which was isolated from food, 2020 from China. Moving on with other varieties of the BLA beta-lactamase resistance genes, with BLA CMA far less than the CSA ones, 129. We did have a MCR 11.1. Looking that particular one up, it's from Michigan in USA, 2024 environmental swab sample, and that's a Cronobacter tersiensis. This one here with a quinolone resistance was from food way back 2014, and that's melanoticus. And then we do have these here, the ATC and the CKO. Unfortunately, this one with the BLA ATC would be of interest because ATC is named after the variety that was initially characterized from Acinetobacter and therefore not an Enterobacteriaceae. Unfortunately, this is one of those where you can see multi-drug efflux systems and there's no assemblies. And similarly, the BLA CKO, there's no assembly available, unfortunately. If we come down to the AMPC, you can see that there's a variety of the BLA genes also occurring with the AMPC. So we've got the AMPC, which is more associated with cephalosporin resistance, and we have the BLA genes as well occurring. Low numbers, but nevertheless found. We also have a MCR variety 11.1. I think that's the second, and these are the only two which occurred. That's again a tersiensis. Again, it's environmental swab sample 2024, USA, Michigan. The final table are the others. Now, this is only an alphabetical issue, so the aminoglycoside resistance genes are A, so taking the AMR at the beginning. There are also the BLA resistance genes in the list here. So please don't think that the initial table of BLA were the only BLA genes that were found, or only AMRs that were found. We are at the tail end. You know, we've got 6 of these, we've got 4 of these, 2, 1, 1, 1, 1, 1. So we are at the tail end of these. Nevertheless, it's interesting to look at the diversity that's occurring. This particular isolate caught my eye because it's got aminoglycoside resistance, it's got beta-lactamases, 1, 2, 3 beta-lactamases, it's got chloramphenicol resistance, trimethoprim, erythromycin, colistin, quinolone, and sulfonamides. And guess what? It's one of those for which there is no assembly available. It's meant to be Cronobacter sakazaki, and it could well be Cronobacter sakazaki. Best I can tell, isolated around about 2014, country unknown. But there's a biosample number. If anyone wants to look it up, investigate further, and if you find the assembly, please let me know, because obviously that is a lot of antibiotic resistance in one strain. Down here, we've got another isolate with aminoglycosides, beta-lactamases, trimethoprim, quinolone, sulfonamides, tetracycline resistance coming in. That's 2012 food isolate China, and that's Cronobacter dublinensis. And then we got this one similarly with quinolones, sulfonamides, tetracycline, etc., with 1, 2, 3 different BLA profiles. And that again is the food one from China, 2020, Cronobacter sakazaki. This one here, you can see again, it's got the multi-drug efflux systems, but again, this one here, there's no assembly available, but there's a biosample if anyone wants to try and look it up and find out a bit more. The final one on our table is simply there happened to be a strain with just one antibiotic resistance, which didn't occur alphabetically or earlier in the tables. So I just looked it up out of curiosity. It's a spice one, 2016 from China, and it's a Cronobacter magensky. So it has no BLA genes, no beta-lactamases in it. So what next? Well, hopefully this presentation may help to guide people to do further research in antibiotic resistance, especially laboratory based, so we can validate and verify and see what the situation is. And in turn, that will hopefully help discussions on the relevance of antibiotic resistance in Cronobacter. It may help to determine if there are trends occurring with antibiotic resistance in Cronobacter. And if you want any follow-up discussions on this, etc., please contact me. My email address is here, and I'd be happy to hear from you. If you want to know more about Cronobacter, then here's a common list of my references. There's many, many more. If you go to my website over here, my email address is here. And who am I? Well, there we are in the last slide. So thank you very much for your attention. I hope you enjoyed it, and best wishes.